low efficacy of drug. So already morning session, Sailaja madam, so given information about linagliptin and their solubility and permeability. So with limited dose only, we are getting the therapeutic activity. So to find out that limited dose, it is highly difficult in general. So because of the low efficacy of the drugs and the drug discovery and development, it is causing so expensive. So for that reason, the simple thing is, for 10,000 synthesized and tested compounds, around 10,000 synthesized and tested compounds will, and lastly we will be getting only one compound that will be approved by the FDA, and that will be coming into the market. So 10,000 compounds synthesized and tested in preclinical studies or development, where only 100 molecules will be in the run, and later in clinical trials maybe 5 molecules or the 5 compounds will be in the clinical trials, and this clinical trials phase is very expensive and longest one. And lastly, if you are fortunate, the researcher is fortunate, like Sailaja madam, if she is fortunate, she will get one molecule maybe after spending 12 to 15 years, and also from the 10,000 library of compounds. And sometimes she may not get that one compound also, but she will lose around 10 years of experience, and also the money and the brain that is invested. So it is a lucky process, and where we have to utilize our brain in a good manner. So out of 10,000 synthesized compounds, lastly approved molecule will be only one molecule. So next, coming to the drug discovery and development process, consists of 3 stages. Next slide please. 3 stages, where it is drug discovery, and later preclinical development, and lastly clinical trials. These are the 3 stages that are involved in the process of drug discovery and development. So drug discovery, finding out a drug molecule, where finding out a hit molecule. What is hit molecule? Hit molecule is the compound which elicits a biological activity, and that hit molecule will be optimized. So once it is having the biological activity, so it is having biological activity, but we have to look after for other parameters, so it should be optimized. The hit molecule will be optimized to optimal affinity, selectivity, and reduced toxicity, and mainly solubility, that is water solubility and lipid solubility and ADME properties. And optimizing all these properties, the hit molecule will convert to form a lead molecule. Further, this lead molecule will be optimized to form a drug molecule. So this is the longest process, where the hit molecule will be identified and optimized to form lead molecule. Further, it will be converted to a drug molecule. So this is in the drug discovery process. That is the first step, or the first process. Later, after finding out the lead molecule, next step is testing that lead molecule in the preclinical development, where the preclinical development will be using animal models, mainly animal models, to find out the safety of the drug, and also some part of efficacy of the drug, and later some of the properties, where we will be in general clarifying the mode of action of drug candidature, clarifying the mode of action of drug candidature or drug, and we will determine some part of pharmacokinetic parameters like bioavailability and drug metabolites or toxic metabolites, or sometimes the routes of excretion, and sometimes the stability of the formulation tests, and these can be studied in the preclinical development. And next stage is clinical stage or clinical trials, which is longest and also so expensive process in clinical trials. And the clinical trials will be in 3 phases in general. The fourth phase will be the post surveillance phase. So first clinical phase, where only 100 healthy volunteers will be involved, and the drug is tested on those 100 healthy volunteers, and everything is available in the textbook and also articles. But so always explanation from a teacher will be always good to understanding the concept. So first phase involves 100 volunteers, healthy volunteers, where what we look forward in that area, we will look forward for the safety of the drug. Safety of the drug on the human being. That is the first phase. In second phase, the several hundreds of the patients will be involved, suffering with the diseases, and drug will be administered to those people, and to evaluate safety of the drug and mainly efficacy of the drug. Efficacy and short-term safety of the drug. And third phase, several thousands of the people are involved. Several thousands of the people involved from different clinical centers of the all over the world. Different clinical centers of the all over the world, and thereby we will be testing, we will be evaluating efficacy and safety and also the side effects of that drug molecule. So last phase will be after releasing the drug into the market, the post-surveillance. Always remember, so after completing the 3 phases also, at any time the drug will be withdrawn from the market if it is showing unwanted effects which are not shown in the clinical trials. So always, totally, every day or every moment, the drug is under the clinical trial phase only, though it is succeeded. So that is about the last stage clinical trials. Please, next slide, please. So the timeline of drug discovery and development, as I said in first slide, the simple thing, here 10,000 compounds are moving to the preclinical stage with our 250 compounds, later 5 compounds in clinical trials, and later, so one compound will be approved by the regulatory authorities. And what is the time it is taking means, around it is 12 years to 15 years. So wasting of 12 to 15 years of time and spending of money and moving to identify a new drug. So lastly, we will achieve only one drug. We don't know whether we will succeed or whether we will fail in that also. So but in general, on an average, the one drug will be approved by using our human brain. We can achieve at least one drug in 10 to 15 years by giving our knowledge. And the cost of the generating this one molecule, it totally arounds on our average 2.6 billion US dollars. 2.6 billion US dollars, and every decade. So this is the indication by, I think, electronic media. That is, I think I am wasting this electronic media also, right? Okay, so wasting of money, wasting of time, wasting of our brain. So if you are unable to achieve at least one drug, then it is totally, the person will be into depression. So, but most of the scientists and researchers, they are actively engaged in the development of the drug molecules, new drugs, and also repurposing the drugs, just like Shailaja madam, and they are improvising the different parameters and moving the drugs into the market to save the lives of the patients. So next slide, please. Next. Okay, simple history. So I don't want to speak about much in depth of the research. In general, Indian civilization is one of the oldest civilization where we are practicing Ayurvedic system, and from starting stage of our ancestors, we used to use the Ayurvedic system where we are collecting the materials from the nature, mainly plant region, mineral region, and mainly plant region. The crude extracts are used to treat the different types of the diseases, and even now the Ayurvedic system is using for untreatable diseases as well as chronic diseases. So like maybe psoriasis or maybe long-term arthritis. So many untreated diseases by or unresolved diseases by allopathic system are treated by the Ayurvedic system. We have such a wonderful formulations in our Ayurvedic system, and you can see. And next one is, though the modern system of medicine or allopathic system is dominating, still we are using our Ayurvedic formulation. So best example, I will give one example, lively example. Till today, for fatty liver or what do you call? Pachakamerlu antaru kada? Right. So doctors are prescribing Live 52 from Himalaya, the company Himalaya. So it is a natural product, Live 52. And nowadays, the doctors are prescribing curcumins. Curcumins isolated from the turmeric powder, pasupu nunchi, turmeric powder. So for fatty liver, some physicians they are prescribing the curcumins and in a formulation way. And other one is, well-known example is to treat the dengue. To treat the dengue, so take one papaya leaf and extract with water and consume that. Automatically your platelet count will be increased. So that is a best example where though the allopathic system is dominating in India. So our Ayurvedic system is always a good one. And next one is, in the history, almost all the discoveries are by the serendipity. Everyone knows about that. This serendipity means by a chance, by accidental one. So in that, the best example is penicillin. The scientist Fleming discovered the penicillin by serendipity, by a chance, where, remember one thing, here I want to give only one thing, that is observation is most important. So while doing the research, keeping your mind or brain and your energies, that is one part, but the best part is observation. So observation gives enormous information where you can keep all those information to bring out something good to mankind. So here also, by the serendipity, penicillins. So next one is one of the chlordiazepoxide, the first benzodiazepine, which was discovered, and later a immunosuppressant drug, that is cyclosporin. Cyclosporin drug, which is scientists are testing for the anti-tubercular activity of cyclosporin. Accidentally, they found that the immunosuppressant activity, and because of that observation, right, immunosuppressant activity, today also the cyclosporin is the first line drug in the organ transplantation. So we are transplanting heart, kidney, liver. So while organ transplantation, what is the drug that is prescribed by the physician or the doctor? That is nothing but cyclosporin. So suppress the immune reactions. If the immune reactions are not suppressed, definitely the person will die. So for that reason, the accidental discovery of this cyclosporin has made some change in the organ transplantation in the human beings. Next slide, please. So next one, after serendipity, next one will be the synthesis. That is nothing but simple chemical modification of the molecules or the compounds, which will provide the therapeutic profiles in an efficient manner. And in that, examples I have quoted here: aspirin. Everyone knows about aspirin, the acetylated product of the salicylic acid. And by the acetylation, why we are acetylating means, right, all the NSAIDs, they have gastric irritation, mainly gastric irritation, so unstable. So to avoid or to overcome these two defects, so aspirin is prepared, and by that there is increase in the stability and reduce the irritation in the gastric mucosa or stomach mucosa. And other example is ranitidine, well-known H2 blocker. So if, right, in general, if you have burning sensation in your stomach, the doctor will always suggest that go for prazoles, pantoprazole, omeprazole, esomeprazole. Already madam, already worked on esomeprazole, right, prazoles before breakfast. But after breakfast, if someone is missing the breakfast, someone is having the breakfast by missing the drug, what is the alternative? So definitely we will go for ranitidine, right? H2 blocker. So here ranitidine, before ranitidine, when we were in M-Pharm, so cimetidine was exciting molecule which was released in the market. Unfortunately, the cimetidine, it has almost 70% of protein binding, and bioavailability is the first drawback of this cimetidine. Later the cimetidine molecule has modified chemically and forming ranitidine, where the imidazole ring in the cimetidine has converted to furan ring in the ranitidine. Because of that, they got higher potency and also prolonged half-life by the ranitidine. So the first sulfonamide drug Prontosil, it is identified or discovered by the virtual screening. The other one, next stage of drug discovery, first chemical modification, and later the next one is virtual screening. By virtual screening, the first sulfonamide was discovered, Prontosil, and later the very exciting drug paclitaxel from the Taxol. So it was discovered by the high throughput screening, where high throughput screening will be used to analyze hundreds of molecules at a time. So it is easy to identify the activity of the 100 molecules at one time, right? So the drug is paclitaxel. Next please. So history, once again, history of drug discovery. I will brief out from the 1919s and also till now. Before 1919, the people are used herbal drugs. Everyone knows about that. So from the plants, the extracts, crude extracts, and that are used to treat the diseases. And like licorice, where the licorice is used to treat the digestive disorders, and sometimes mint is used as mouthwash and also as a toothpaste like. And serendipitous discoveries, already I said that penicillin is the best example, cyclosporins. And in 1920s and 1930s, so the discovery of vitamins and vaccines, vitamins D, E, K were discovered in 2020s and 30s, and vaccines in 1923, the first vaccine, not first vaccine, it is a diphtheria vaccine. Later in 1924, scarlet fever vaccine, and everyone knows about tetanus, right? Tetanus. So if you are with interacting with any iron material or steel material which is having a rust, definitely will go for TT, tetanus toxoid. So it was discovered in 1924. So development of the vaccines in 1940s, and it is known as antibiotic era. In this antibiotic era, penicillins, mainly penicillins and also penicillin derivatives, and 1940 penicillins, 1944 aminoglycosides, and 1948 tetracyclines. So penicillins, the usage of penicillin and penicillin derivatives was excellent during the World War II. So millions of soldiers saved by this penicillin and also penicillin derivatives. Because of that reason, scientists worked hardly to develop antibiotics, and later they developed aminoglycosides and also tetracyclines. And here to say the tetracyclines, our own person from this Andhra Pradesh, Yallapragada Subbarao garu, yes, from the BHIMAVARAM, Yallapragada Subbarao garu, he was the person to identify the tetracyclines. He discovered 20 tetracyclines, and he was the person to identify the pathway of the cancer and even anti-metabolites, methotrexate. All these are due to by the great personality, that is Yallapragada Subbarao garu. We are always thankful to him, and we should be always show gratitude to the Yallapragada Subbarao sir. And in 1950s, new technologies were discovery of DNA, that is genome, discovery of genome, DNA material. The Watson and also Francis scientists, they discovered DNA. And later in 1960s, breakthrough in etiology, that is related to the disease, pathway of disease, formation of the disease, the etiology. Because of the etiology, the scientists discovered estrogens. And later in 1970s, rise of biotechnology, everyone knows that biotechnology gives always monoclonal antibodies and also vaccines. In corona period or COVID-19, we are all dependent on the vaccine, mainly vaccine, COVID vaccine. So use of IT started in 1970s, and next in 1980s, commercialization of drug discovery, combinatorial chemistry, where library of compounds were synthesized and they are tested. And unfortunately, this combinatorial chemistry was not succeeded that extent. And later in 1990s, and also now robotics and automation. So all the softwares and automation means the best example now is Adam and Eve. Robots of Adam and Eve, they are using in the artificial intelligence. And this is the simple brief history about the drugs that are started from 90s to till now. Next slide, please. Brother. Okay. Drug discovery methods. So if I am boring, I will stop here. Shall I continue? Right, okay, thank you. So what are the drug discovery methods? Already I explained about serendipity, by a chance, by accidental process, penicillins and cyclosporin. Next one is random screening. Earlier, before finding of these chemicals and also the penicillins, random screening by the folkloric medicine. That means depending upon the villages that they are using the plants and also parts of the plants in the treatment of different diseases and ailments. Depending upon that information, the people collected, the scientists collected medicinal plants. Later they have extracted and they have tested, screened for the pharmacological activities, and once they are proved, and later they have gone for the isolation of the active constituent in that plant extract. So random screening of the medicinal plants. Even random screening is also applicable in the synthesized compounds also. Next one is chemical modifications. I already explained with a simple example, aspirin and also ranitidine. And high throughput screening. By high throughput screening, already given an example, that is the well-known example, anti-cancer agent from the natural region, that is paclitaxel, so which is discovered through this high throughput screening. And later the most used method nowadays is rational drug design. It is a rational drug design, and this rational drug design is a smartest one and cheapest approach. Smartest and also cheapest approach of this rational drug design. So I will speak 5 minutes about this rational drug design, because if anyone is interested, so this is the process, how to start the discovery of the molecule or the researching a new molecule. So in this rational drug design, it starts from the finding of the target. So here started finding of the target. First time before that, people thought that only a ligand or a drug molecule, a compound, which will relieve the symptoms. But when the rational drug design started, so they thought about the target molecule. What is the target molecule here? The target molecule is a bio-macromolecule. What are the bio-macromolecules? Mainly proteins. So bio-macromolecule, and to this target, so we will find out the hit molecule which will interact or which will bind to the bio-macromolecule or to the protein. So therefore, it will become, if it is having a binding affinity, a sufficient binding affinity, definitely it shows pharmacological activity. So that will become a lead molecule. So that hit molecule will be optimized in all the parameters. As I said, in affinity, optimal affinity, selectivity, non-toxicity, and also ADME factors and ADME properties and everything. So after optimizing all this, it will become a lead molecule. And in this lead, if you then generate this lead molecule, definitely it will bind to the macromolecule and it will elicit its pharmacological action. So in this connection, here the rational drug design is divided into two approaches. One approach is ligand based, where you can listen carefully here, ligand based, and other one is structure based. Ligand based means where the target molecule is unknown, where the protein or the macromolecule is unknown. So for that reason we will design the ligand with some sort of predictions, and we will interact this ligand with the macromolecule. That is ligand approach, ligand design approach. Another one is structure based approach, where the target macromolecule is a known one. That means 3D structure of protein is known. Once the structure is known, we can interact the areas of the protein or the sites of the protein with our designed ligand. That ligand will be a complementary one to bind to that the known 3D protein structure, right? So known one. So in these two approaches or two ways, one can move towards the discovery of a new drug. So this is about the new approach. Later several things happened, that is the target molecule is macromolecule, and once it is a macromolecule, this is involved in the disease causing one. So disease causing one, once it is disease causing one, if we are able to change that the in the macromolecule where it is malnutrition or it is deficit, if you are able to change that macromolecule with that of ligand, definitely we will change the etiology of the disease, pathophysiology of the disease, and symptoms of a particular disease. Then comes what is that macromolecule. The macromolecules are the generally proteins. In general, 20,000 of protein genes, sir, 20,000 of protein coded genes are present in human genome. Remember this, human genome consists of 20,000 protein coded genes. So among those, 3,000 will be established or estimated as druggable targets, or druggable genomes or druggable targets. That means we have 3,000 targets. We have 3,000 targets in one genome. So that means we have many opportunities to hit that one. So therefore it will be always useful. And what is that macromolecule means? Proteins. And this human protein, it is almost 53% in the genome, sorry, in the body, and among that 38% of human protein is not studied. That means the unstudied proteins are 38%. That means there is huge gap or huge area where you can work. You can work to identify the structure of the protein and also give that protein structure for finding out the new drug molecules, right? Okay, after that, so this is a lengthy process where that has been generated to formation of the later the structures of the proteins were confirmed or identified by X-ray crystallography and also NMR spectroscopy, and later cryogenic electronic microscopy, and they have collected almost 180,000 of proteins. 180,000 of proteins. Remember, 180,000 of proteins are stored in a PDB, protein data bank, and all are available in the online freely. You can take at least one protein and you can check that protein with the different ligands, and where it requires only laptop or the desktop or a mobile also. So you can check find new molecules with that of a protein structure. So after the development of that, so later the molecules are calculation of the properties moved into mathematical ones, right? Measurements. So where they are development of QSR, CAD, so where the molecule or the structure of the molecule is encoded, and once the molecule is encoded, and that can be compared with the available drugs, and after molecule is encoded, and definitely the molecular properties of the molecule also should be encoded, and they are called in the form of descriptors. So depending upon the structure of the molecule, 1D structure, 2D structure, 3D structure. So 1D structure, from the 1D structure, you will be getting the only element composition, mainly element composition and molecular weight in the form of descriptors, and 2D descriptors, 2D structure, and they will be generated 2D descriptors. These descriptors in the form of the amount of, that is, hydrogen donors and also hydrogen bond acceptors. Hydrogen bond, hydrogen, always hydrogen bond plays very important role in binding of the ligand to the target molecule. Maybe target molecule may be protein, or maybe a gene, or anything else, or receptor, or enzyme, anything. So this hydrogen bonding plays very vital role in binding to the target molecule or the target. So here descriptors, 2D descriptors, where we will be observing the hydrogen bond acceptors and also hydrogen bond donors, acceptors or donors, and later 3D structure, which will speak about the molecular surface area and general surface area of the molecule, and 4D structure with all descriptors. So I think it is boring, so it will be it will be giving the information about protein and binding sites of the target molecule. Okay. So later generation of the structure activity relationship, QSR, so many things were happened, but I will give only one example, a lively example. When I was doing my M.Pharm and PhD, I am a natural product chemist. I collected medicinal plants, extracted, and later isolated the compounds, and later structure illustration by different spectroscopic techniques, and later tested for the activity. This is one type of research. And synthesizing the compounds and identifying the structures by the spectroscopic techniques like UV, IR, NMR, 2D NMR techniques, and later activity of the molecule. This is in chemistry. So madam will be doing after that. After finding out, she will be doing formulating that one, and solubility, permeability, and further bioavailability. So later your principal, the KGRL college principal, Dr. Raghava, he is my student. When he is doing his PhD, we designed according to rational drug design. I did my PhD in 2005 to 2018, 8, and Raghava registered in 2011 when my daughter's born. So he started in 2011. So that means from 2005 to 2010, so the scenario has changed. So rational drug design. So now we prepared a scaffold. How to do the research means find out the natural product, biologically active natural product. Go through the literature. Find out the natural product. Modify slightly. We have taken luteolin, and we have converted that luteolin, simple 6-membered ring is converted to 5-membered ring, a furan ring. Right? Simple change in the molecule, and we have taken that furan, benzofuran, and after that he has prepared 325 compounds, series of compounds by using CAD, by using computer aided drug design. When I was working, I don't have that computer aided drug design, but he has that advantage. He has prepared 325 molecules by using the CAD software. After that, by virtual screening, he screened that one and identified some affinity, and later he did docking. So now docking is, I think in your college, Devi is also working on docking. She is well expertise in the docking. So 2 people are in your college, sir. So simple thing is, by doing docking, by using laptop, simple software, we will get many number of publications, sir. So this is dry lab. So no need of laboratory. Just like madam is working in laboratory, we don't require all those things. We require only laptop and software. That is enough. We will get number of hundreds of papers. So after docking, so he screened some compounds. Lastly, using all these tools, all the software tools, lastly he find out 30 molecules are active by using physicochemical properties by the QSR studies, by using Hans analysis. So Hans is the a father of drug design. So he tested in 1964 penicillin derivatives, antimicrobial activity penicillin derivatives correlating with the descriptors, and he succeeded in that. So he was considered to be as a father of drug design. Hansch, even M.Pharm students, they used to study Hansch analysis. So by using QSR studies and some visual screening, high throughput screening, and keeping all the tools, he lastly selected 30 molecules, and he synthesized 30 molecules and later checked for the anti-cancer activity, and he succeeded in that. Out of 30 molecules, only 6 molecules are showing promising activity. Only promising activity. So to complete the PhD thesis, and it has taken almost 3 years to complete this. Though he is using advanced tools, so I think it is boring. Brother, where is my slide? So that is very simple thing, where one can design the drug molecules by yourself. So I will complete within 5 minutes, last 5 minutes. So this is already explained by our associate professor, Shailaja madam. So target selection is the most important thing always. For earlier times, cellular, that is cells. Antibiotics acts always on the cell membrane, destroying the cell membrane, cells. And next developed one is genomics, acting on genes, and later proteomics. Today the proteins are playing a crucial role for finding out the new drug molecule. So proteomics, and lastly bioinformatics is the area where the complete data will be analyzed by the software tool. So rest of things are common things. Next slide please. Next. Well, and chapter next. Next. So future prospects, everyone knows about artificial intelligence. Even chai wala, pan wala, everyone is saying about artificial intelligence. Recently one video went viral in the WhatsApp, where one lady is giving news. That is a robo lady giving the news. So that is created by the artificial intelligence. Here in simple, artificial intelligence and machine learning, deep learning. So artificial intelligence is nothing but a machine performing just like human brain, mimicking the human brain by the machine. So even machine learning is nothing but by using algorithms, mathematical calculations, algorithms, and pooling of the data and making out the new drugs or the parameters in a easy manner. Deep learning is nothing but it is in depth of the artificial intelligence, the deep learning where neuronal networks are used to predict the actions of the drug molecule. Next please. So what is artificial intelligence? Everyone knows that simulation of the human intelligence processed by the computers. The computers are acting like our brain, so that is artificial intelligence. So several areas we are using, educational system and also business. Next please. Next slide please. Brother, next. So this is the picture which will give simple information, diagrammatic representation, where the artificial intelligence is mimicking the brain. Next please. Next please. Applications of artificial intelligence. So I will complete within 3 minutes. So artificial intelligence, so it is a versatile tool that will be used to used in the drug discovery as well as drug design and also drug development. And in that, identification and validation of drug targets like proteins or genes, designing of a new drugs, and prediction of the 3D structures of the proteins. Next, drug protein interactions or drug interactions, and drug repurposing. After corona, there is great need in the drug repurposing. Already we have lakhs of drugs in the market, so not lakhs, thousands of drugs in the market. So using those drugs, repurposing, right? Using those drugs for and testing for different diseases. So drugs repurposing. Next slide please. So artificial intelligence, so it is a versatile tool that will be used to develop, use in the drug discovery as well as drug design and also drug development. And in that, identification and validation of drug targets like proteins or genes, designing of a new drugs, and prediction of the 3D structures of the proteins. So understanding the pathways or mechanisms, and finding the molecular targets, and finding the lead molecules or the hit molecules, and synthesis, which is used in the synthesis routes, synthesis planning in a different synthetic routes, and predicting mode of action or mechanism of action, selection of population in the clinical trials, and pharmacovigilance also, and polypharmacology, and clarifying in reaction mechanisms, so predicting ADME factors. So artificial intelligence is involved in everywhere, everywhere. Next slide, please, brother. So I will give only one example. There are many examples. So AI platforms. Now all the companies, all the pharma companies are keeping tie-up with or investing money with CROs who are developing AI, artificial intelligence. So to find out, in that connection, the one example here it is, the popular AI platforms is the Swiss Drug Design. Swiss Drug Design, and this is open one. You can see even in online. It is free. So open one where we can see that Swiss Drug Design, it involves molecular docking and pharmacokinetics and drug likeness prediction, virtual screening, lead optimization, target prediction of the small molecules. Like this, several companies, AstraZeneca and even Bayer, all the companies, they are invested their money on the CROs who are developing this artificial intelligence. Some people, they want to know how the small molecules will bind to the proteins, and some people, they want to find out different synthetic routes, many synthetic routes where we can find the drug molecule. Like that in all the research areas, even how to select the population in clinical trials, that is a difficult task. For that also even the AI platform is used. And lastly, I will say only one thing. For Raghava, he spent 3 years of time to find out 5 molecules, but by artificial intelligence, hardly within 3 months, you can complete and you can find out minimum 3 to 5 compounds in a significant activity, right? So thank you very much for listening very patiently. Thank you. Sorry for the boring lecture. And lastly, I want to thank our director. So not young, but he is young and dynamic. Our Nageshwar sir, Dr. Nageshwar sir, always he is young in mind, so he is active. Sir, thank you, sir. Thank you for giving this opportunity.
Thank you, sir, for your enriched knowledge. Now I request Chairperson M. Venugaru to give feedback about this session.
Good evening to all the dignitaries. And sir share a very good explanation about the drug discovery. Basically, I am the background of pharmaceutical technology. So my studies are properties of drug and properties of excipients. Excipients and drug plays a very important role. So drug discovery. So today sir deal with drug discovery and rationalized drug design, drug docking techniques, and history of the drug discovery. So today we are gained very important knowledge about the drug discovery, and sir told that importance of pharmaceutical chemistry person, pharmacology person, and pharmaceutics person. So these all are plays a very important role, and a very good explanation, sir. Thank you so much.
Now I request Co-Chairperson Y. Varadaraju Garu to give feedback about this session.
Thank you for giving a wonderful opportunity. Thank you, sir, for giving a wonderful presentation about intellectuality properties of drug discovery sections. And thank you very much, sir. Thank you.
Now it's time to felicitation for guest speaker, chairperson and co-chairperson. I request respected director sir, principal sir to come on to the dais to felicitate the guest speaker, Dr. G. Sridhar Babu Garu. Thank you, sir, for your acceptance. Now it's time to felicitate our chairperson, M. Venu Garu, Assistant Professor, Lydia College of Pharmacy. I request Director Sir and Principal Sir to felicitate him. Thank you, sir, for your acceptance. Now it is felicitation for co-chairperson Y. Varadaraju Garu, Assistant Professor, Nova College of Pharmacy. I request Principal Sir and Director Sir to felicitate him.
So it is hope all are awaiting your results about poster presentations, e-posters. What else? Indian passport, sir? Warrants. Okay. So shortly it will be announced. In the meantime, I wanted to have a dialogue or interaction with you all people. Hope you are all happy and enjoyed a lot and learned a good knowledge about scientific sessions. ఎవరికైనా ఏమైనా ఇబ్బందులు కలిగితే జస్ట్ ఎక్స్క్యూజెస్ బికాజ్ ఎందుకంటే టుడే ఓవర్వెల్మింగ్ రెస్పాన్స్ వాట్ వై ఎక్స్పెక్టెడ్ మోర్ దెన్ దట్ యూ హ్యావ్ కమ్ అంటే స్పాట్ అందుకనే ఫర్ ఎనీథింగ్ ఆర్గనైజేషన్ ఈవెంట్ వీ మస్ట్ హ్యావ్ సమ్ ప్లానింగ్ సో ఫర్ ద లాస్ట్ 3 days ఐఎమ్ ఆస్కింగ్ వాట్ ఇస్ ద ఎక్స్పెక్టెడ్ నెంబర్. So all of a sudden today spot registrations maybe around 150. So it is not accounted actually. So for that food is little bit less shortage vachindante. So for that one should have a master planning. So today we are conducting andukane prati daniki there will be a fixed date. So 9th in the function ante 4th was the last date. You have to give feedback or how many number you are attending. So we should have some planning. Suppose I have planned for 500 members. Today I think about 600 people have come. So for 100 members we are not planned. So ultimately shortage vastundi food. Manaki migilte problem లేదు కానీ తగిలితే ఇస్ ఏ పించింగ్ ఐటెం. సో ఎవరికైనా మీ అందరికీ బహుశా హోప్ యూ ఆల్ అవుట్ సైడ్ స్టూడెంట్స్ హ్యావ్ ఎంజాయ్డ్ యువర్ ఫుడ్, డెలిషియస్ ఫుడ్. బాగుందా లేదా? బాగోలేని వాళ్ళు చెప్పండి, బాగోలేదన్న వాళ్ళు. ఏదమ్మా? ఎక్సలెంట్, వెరీ గుడ్, థ్యాంక్ యూ. సో వైల్ గోయింగ్ ఐ థింక్ ఫీడ్బ్యాక్ ఫార్మ్స్ పెట్టారా? ఫీడ్బ్యాక్. సో ఎనీ ఈవెంట్ యూ కండక్ట్ యూ హ్యావ్ టు గివ్ యువర్ ఫీడ్బ్యాక్. సో ఇఫ్ యూ గివ్ ఫీడ్బ్యాక్ దట్ ఎక్సలెంట్, సో హ్యాపీ. బట్ ఐ వాజ్ నాట్ హ్యాపీ బికాస్ ఐ హర్డ్ దట్ ఫుడ్ ఈజ్ షార్టేజ్. సో ఫర్ దట్ ఐఎమ్ asking for the excuse. Okay. ఈ లెవెల్ అడగక్కర్లేదు, బట్ సో ఇట్ ఈస్ హ్యాపెన్. లాస్ట్ టైం ఫుడ్ మిగిలిపోయింది, సో we sent to the hostels. ప్లానింగ్. సో నేను ఎంతమంది వస్తున్నారు ఏంటనేది అనుకోండి. సో అకార్డింగ్ take your certificates. That last moment it is coming out. So all are eagerly awaiting for that one only. ఏ కాలేజీ వాళ్ళకి వచ్చింది, ఎవరికి వచ్చింది. సో ఎక్సలెన్సీ అమాంగ్ యూజర్స్ హూ ఇస్ ఎక్సెల్డ్ వెల్ వస్తుంది. సో లెట్ us హ్యావ్ ఇట్, ఎంజాయ్ చేసి. సో జాగ్రత్తగా మీరు అందరూ వచ్చినందుకు వన్స్ అగైన్ మనకు ఆపర్చునిటీ ఇస్తారో లేదో తెలియదు, బట్ ఐ హావ్ టేకెన్ ద ఆపర్చునిటీ బికాస్ టైమ్ ఈస్ దేర్. సో ఐ విష్ యూ ఆల్ ది బెస్ట్. Have a nice time. Thanks for coming over here and try to attend as many seminars as possible. It creates for you credibility will be increased. You will be learning a lot. మీరు వచ్చారు కాబట్టి తెలుస్తుంది. సో మేడం వచ్చారు, శ్రీధర్ బాబు వచ్చారు. సో లైక్ దట్ అటెండ్ యాజ్ మెనీ యాజ్ పాసిబుల్. Every college is organizing. So try to, because maybe spending 200 rupees or 300 rupees, it is doesn't matter. But you will not get. Suppose tomorrow you wanted to go for BPharma outgoing students. So tomorrow you wanted to go for PG programs. Which college better? Now I am happy to announce that M.Pharm admissions are over. In Andhra Pradesh, KGRL College of Pharmacy, 100% seats are filled. 100 seats, 100% seats are filled. No vacancy. So people are in the queue. I want seat in a drug regulator base. I'm sorry, even if you pay 10 lakhs rupees, I cannot offer the seat. So like for that, we have struggled a lot, built up the image. To get a good name, it takes years. To get a bad name, it's a matter of seconds. So today, I'm happy to announce that KGRL Pharmacy, all M.Pharm seats are 100% filled. People are in the queue, but we are not in a now today will be completing the process. So only vacancy EWS, economically weaker sections. Our economically weaker sections ki one should have economically weaker section certificate. It will be issued by the revenue department for 23-24 academic year. అటువంటి వాళ్ళు ఎవరిని ఉంటే there are some two, that is extra given seats. 15 full. Only 2 seats. EWS, economically weaker section quota is there. So for that, students must have some awareness. EWS అంటే ఏంటి? ఇప్పుడు 2 lakhs లోపల వైట్ రేషన్ కార్డు ఉన్న వాళ్ళకి అక్కర్లేదు. EWS సర్టిఫికేట్ అంటే అప్ టు 8 lakhs యూ కెన్ హ్యావ్ ఇన్కమ్, ఫ్యామిలీ ఇన్కమ్ గ్రూప్. అందులో EWS సర్టిఫికేట్ తీసుకుంటే యూ ఆర్ ఎంటైటిల్ టు గెట్ ద రిజర్వేషన్ అండర్ EWS రిజర్వేషన్, యూ విల్ గెట్ ద సీట్. అది ఒక్క ఓన్లీ EWS వాళ్ళకి. ఎనీ ఓసీ పీపుల్, కాపు, కమ్మ, వైశ్యాస్, క్షత్రియాస్, all people they can have it, but provided they should have income of less than 8 lakhs and some parameters are there. ఇంత ల్యాండ్ ఉండకూడదు, ఇంత హౌస్ సైట్లో ఉండకూడదు అని చెప్పేసి ఉన్నాయి. EWS సర్టిఫికేట్ తెలుసా అందరికీ? ఓకేనా? సో ఇప్పుడు అవుట్ గోయింగ్ స్టూడెంట్స్ ఉంటే ట్రై టు హ్యావ్ సర్టిఫికేట్స్ ఇన్ యువర్ హ్యాండ్. సో 24-25 ఇప్పుడు ఫైనల్ ఇయర్ ఉన్న వాళ్ళకి 24-25 academic sessionకి you will be joining. So for that you keep ready and you can avail the reservation under the EWS. Okay, thank you very much. So all faculty members, those who have come from other places and acted as chairperson, co-chairperson, thank you once again, and see that you did a good job. So let us encourage that. Thank you very much. Okay, once again thank Shailaja madam. So Shailaja madam, she will be upcoming professor and definitely she will have a bright future. Definitely she will be having a bright future. So I will extend my cooperation because andarki chestam. So my helping nature is I communicate each and everything, whatever the knowledge I get, I communicate to each and everyone. Whether my student or my faculty or not like that. Total, universally, actually I should not say about myself, but I do my job on universal basis. Any communication, Nava College వాళ్ళు ఉన్నారు ఇక్కడ. Now this for the last 2 days, the last night 24 hours నుంచి we are working on the abstract counseling. So colleges, PCA approvals for 23-24. చాలా స్ట్రగుల్స్లో లాస్ట్ నైట్ 6 o'clock we got the approvals from PCI. వచ్చిన అప్రూవల్స్ వీ హ్యావ్ టు సబ్మిట్ ద స్టేట్ టెక్నికల్ ఎడ్యుకేషన్. మీ పేరు కూడా ఉందో లేదని ఇంకా సబ్మిట్ చేయలేదు, జస్ట్ ఫాలోఅప్ విత్ యువర్ మేనేజ్మెంట్. ఓకే, సో there are about 10 colleges are there which have not submitted their PCI approvals. So we are working on the state level. So communication, మా మేడం విజయరత్న మేడం మాకు జూనియర్. So she has given my title, as long as Nageswar is there, he is a PRO for pharmacy, profession in India, all India level, PRO for pharmacy. So whatever communications, whatever knowledge we get will be communicated to all people. So same message I forward to all my students, first year, second year, third year, fourth year, M.Pharma, each group will be there. So any important message I will communicate, and I put the message in my status also. So many people they will see and revert back to me, so how to do it. So like that I do my service. So thank you very much for giving this opportunity. So let us progress.
Thank you, sir.
Good evening, everyone. Hello. So being the principal, so I am giving the conclusion note. So first of all, I thank all the college students, staff members, principals and the management for encouraging and sending their students to the international seminar conducted at KGRL College of Pharmacy. First of all, I thank everyone. So being this report, so the list of students attended from various colleges. Vishnu College of Pharmacy, BHIMAVARAM, there are 22 students from the college. Vasavi College, Tadepalligudam, there are 10 students from that. Lydia College, 60 students from the college. CSN College of Pharmacy, 17 students. AU College of Pharmacy, 5 students. AKRG College of Pharmacy, 5 students. Nova College, 48 students. VVIPS, Sheikh Rashid is the staff member. 22 students are awarded. And surprisingly from AKNU, 32 students are awarded. And Paida College, 33 students. JITS College, 17 students. And our college, about 270 students. On an average, a total of 540 students attended today's seminar. So now I have given the number. So I need 2 minutes of your attention. So you have your friends with you for your next coming years. But I can't talk to you for the next coming years. Please spare your 2 minutes to me. Give me your 2 minutes of time. Okay? So I want to tell you one thing. This is not only the numbers. So today we have interacted with various teachers. Now I am happy to say I am a first year student for Shailaja Madam in Talapadmavati College of Pharmacy. And a 4-year student, first year to final year student for Sridhar Babu garu. And he is my PhD guide and my well-wisher. Nenu em cheyalanna on my professional or personal or anything. First I will discuss with the sir and Nageswar garu. So both the pillars are available with me today. So, ఇది మీకెందుకు చెప్తున్నానంటే you select your own teachers in the institution. they will guide you till the end of your life, not for fun. ఒక స్టూడెంట్ హ్యాపీగా డెవలప్ అయితే దానికన్నా ఇంకోటి ఏమీ టీచర్కి అక్కర్లేదు. సో సెలెక్ట్ యువర్ టీచర్స్. మా స్టూడెంట్స్కి ఎప్పుడూ చెప్తాను నేను, నాట్ ఇన్ ద కాన్సెప్ట్ ఆఫ్ సబ్జెక్ట్, దే విల్ గైడ్ యూ ఇన్ ద కెరీర్, లైఫ్ అండ్ ద పర్సనల్ థింగ్స్. ఏ టీచర్ ఈజ్ అ టీచర్ టిల్ యువర్ డెత్. ఒక్కసారి మీకు టీచర్గా వస్తే మీరు చనిపోయేదాకా వాళ్ళు టీచరే. ఇవాళ నేను టీచర్ అయ్యాను, ప్రిన్సిపల్ అయ్యాను, నాకు ఆయన టీచరే. నేను డైరెక్టర్ గారి దగ్గర స్టూడెంట్గా జాయిన్ అయ్యాను. ఎంఫార్మసీలో నేను సార్ దగ్గర స్టూడెంట్ని. నన్ను తీసుకొచ్చి ఇక్కడ లెక్చరర్ని చేశారు, అసిస్టెంట్ ప్రొఫెసర్ని చేశారు, అసోసియేట్ ప్రొఫెసర్ని చేశారు, ప్రొఫెసర్ని చేశారు, నవ్వు ప్రిన్సిపల్ని చేశారు. అయినా నేను ఆయనకి స్టూడెంట్నే. సో అన్ని కాలేజ్ స్టూడెంట్స్ ఉన్నారు కాబట్టి చెప్తున్నాను, సెలెక్ట్ యువర్ టీచర్ ఇన్ ద ఇన్స్టిట్యూషన్, ఫాలో దేర్ గైడ్ లైన్స్. Definitely a teacher will guide you in a proper way. No teacher will guide you in a wrong way. Why? Because there is no loss and profit in this. So every teacher will be happy when their students will be settled in good position. So మాకు ఇంక్రిమెంట్ వస్తానో ఏదో వస్తే హ్యాపీ కాదు. సార్ నేను పలానా చోట పలానా కంపెనీలో జిఎంగా సెటిల్ అయ్యాను, సైంటిస్ట్గా ఉన్నాను అని చెప్పిన రోజు మాకు ఎంతో హ్యాపీగా ఉంటుంది. ఇంకొక ఎగ్జాంపుల్ చెప్తాను మీకు. ఇవాళ ఆన్లైన్లో నరేందర్ రెడ్డి అని ప్రిన్సిపల్ సైంటిస్ట్ వచ్చారు, కానీ మేమిద్దరం ఆకతాయిగా సార్ దగ్గర తిరిగాం. అదే స్టూడెంట్ ఇక్కడ నేను ప్రిన్సిపల్గా ఉన్నాను, అతను ప్రిన్సిపల్ సైంటిస్ట్గా ఉన్నాడు. ఆ సార్కు ఉండే ఆనందం బయటికి చెప్పలేకపోతున్నారు. ఐ యూస్ టు ఎక్స్ప్రెస్ విత్ అస్. అలాగే డైరెక్టర్ గారు ఇక్కడే ఉన్నారు, నిర్మలా మేడం. ఎప్పుడో 2009లో మా అందరికన్నా ముందు వచ్చారు. సో ఆవిడ్ని అసిస్టెంట్ ప్రొఫెసర్గా, అసోసియేట్ ప్రొఫెసర్గా దగ్గరుండి పిహెచ్డి రిజిస్ట్రేషన్ చేయించి, సబ్మిషన్ చేయించి, ఎవరు ఫాలో అప్ చేయరు. పిహెచ్డి ఎంతవరకు వచ్చింది, ఎంత చేశారు, సబ్మిషన్ అయ్యిందా, ఏమైనా ఇబ్బందులు ఉన్నాయని ఎవరు ఫాలో అప్ చేయరు. ఓన్లీ నాగేశ్వరరావు గారే చేస్తారు. ఒక స్టాఫ్ మెంబర్ తీసి సబ్మిషన్కి వెళ్తే అక్కడ యూనివర్సిటీలో ఏమో ఇబ్బంది వస్తుందో ఏంటో అని కంగారు పడిన ఏకైక పర్సన్ మా డైరెక్టర్ గారు. దానికి కావాల్సిన ఏర్పాట్లు ఏమీ రాకుండా, ఇబ్బందులు ఏమి రాకుండా చూసుకున్నారు. సో ఇలాంటి టీచర్స్ ఉంటారు, అది మిమ్మల్ని చూసుకోమంటున్నారు. రెండు, సో మోస్ట్ ఆఫ్ ద బీఫార్మ్ స్టూడెంట్స్ ఆర్ విల్లింగ్ టు రికార్డ్, దిస్ ఈజ్ ఓన్లీ జనరల్ సజెషన్, మోస్ట్ ఆఫ్ ద బీఫార్మ్ స్టూడెంట్స్ ఆర్ నాట్ ప్రిపేరింగ్ ఫర్ ద మాస్టర్స్. మాస్టర్స్ సీట్స్ మీకు అవైలబుల్గా దొరుకుతున్నాయి. సో మీరేమి పీజీ సెట్లు రాయకుండా యు ఆర్ వెయిటింగ్ ఫర్ ద సీట్స్. మీరు మాస్టర్స్ చేయాలంటే యు రైట్ యువర్ క్వాలిఫైయింగ్ ఎగ్జామినేషన్, సెలెక్ట్ యువర్ సీట్ ఇన్ ఏ ప్రాపర్ ఇన్స్టిట్యూషన్, డూ ద కోర్స్ ఫర్ ద 2 years. ఇవాళ ఉన్న స్టూడెంట్స్ అందరూ బీఫామ్ అయిపోవాలి, నేను సంపాదించేయాలన్న కాన్సెప్ట్లో ఉంటున్నారు. సంపాదించండి. 2 ఏళ్ళు నాలెడ్జ్ సంపాదిస్తే మీరు బీఫామ్తో సంపాదించేదానికి 4 times సంపాదిస్తారు. సో ప్లీజ్ వెయిట్ ఫర్ 2 more years అండ్ రీడ్ వెల్ for the MPharm courses. So the upcoming drawback ఏంటంటే most of the MPharm students are not going or not willing to continue their academics properly. ఏదో కోర్స్ కంప్లీట్ చేయాలన్న ఇంటెన్షన్తో ఉంటున్నారు మీరు అందరూ. సో సంపాదన కోసం ఒక 2 ఏళ్ళు ఆగండి, 2 ఏళ్ళు నేర్చుకోండి. అప్పుడు నేర్చుకున్నది మీకు లైఫ్ అంతా ఉపయోగపడుతుంది. అప్పుడు మిస్ అయ్యారా మీకు లైఫ్లో ఎక్కడా నేర్చుకోలేరు. సో మాస్టర్స్ ఈజ్ ఏ డివైన్ కోర్స్. మీరు అందరూ ఫైనల్ ఇయర్లో ఉన్నారు కాబట్టి ఇది ఒకటే చెప్తున్నాను. ఓకే, లర్న్ యువర్ మాస్టర్స్ కోర్స్ పర్ఫెక్ట్లీ, బీ ఏ గుడ్ ప్రొఫెషనల్. సో థ్యాంక్ యూ వన్ అండ్ ఆల్. థ్యాంక్ యూ ఆల్ ద స్టూడెంట్స్ ఫ్రమ్ all the institutions, and I thank all the organizers of KGRL College of Pharmacy, and especially thank the co-convener B. Shweta, where is she? So for organizing this event, and I thank all the teaching staff members for coordinating this event, and not only teaching staff, non-teaching staff, all the organizers, I thank you once again. Now we'll move to the felicitation ceremony of Dr. K. Nirmala, who has been awarded doctorate in the last day. So I request K. Nirmala garu onto the stage.
అది కూర్చోండి. వన్స్ అగైన్ బికాజ్ ప్రెస్ వాళ్ళు కూడా వచ్చారు. అదృష్టం అంటే ఎలా కలిసి వస్తుందో చూడండి. ఓకే, ప్రెస్ వాళ్ళు బాబీ గారు చాలా మంచి రిపోర్టర్, ఎలక్ట్రానిక్ మీడియా, కరెక్ట్గా అనుకున్న టైంకి భగవంతుడు ఆయన పంపించారు ఇక్కడికి. సో నిర్మలా మేడంకి ఎస్టర్డే ఓన్లీ షీ హాజ్ రిసీవ్డ్ హర్ PhD award from Andhra University. Okay, so she is the regular faculty member for the last 10 years, 14 years. సో ఆ అమ్మాయికి పర్సుయేషను అంతా బ్రహ్మాండంగా జరిగింది. సో అనుకోకుండా మన సెవెంత్ని ఆంధ్ర యూనివర్సిటీలో అవార్డు అవ్వడము, దానికి కావాల్సిన ఏర్పాట్లు అంతా అన్న అమ్మాయి సంపాదించుకుంది, కష్టపడి చేసింది, అవార్డు వచ్చింది. మనకి ఈరోజు ఇంటర్నేషనల్ సెమినార్ పెట్టుకున్నాము. సో అది ఇది కలిసి వచ్చింది కాబట్టి టుడే వీ వాంటెడ్ టు సెలబ్రేట్ ఆర్ ఫెలిసిటేట్ డాక్టర్ నిర్మలా ఆన్ దిస్ అకేషన్ because she is she has already received awarded PhD మొన్న అయింది, వైవాస్ అయింది. ఆ మేడం కూడా స్టూడెంట్స్గా ఉన్నారు. సో ఈ సందర్భంగా మీ అందరి సమక్షంలో KGRL కాలేజ్ ఆఫ్ ఫార్మసీ తరపున, నా తరపున, ఫ్యాకల్టీ తరపున, అందరి తరపున నిర్మలాని డాక్టర్ నిర్మలా చేస్తూ షీ విల్ బి ద ప్రొఫెసర్ and she will be elevated. Only ఇప్పుడు దాకా now she as a professor, associate professor, she is drawing a salary of less than maybe 25,000 or 30,000. So award, PhD award తీసుకుంది. So she will be elevated and with higher scale. So maybe అంటే ఫిగర్ అది మేనేజ్మెంట్ చెప్పాలి. సో డెఫినెట్లీ PhDకి M.Pharmకి PhDకి ఎంత తేడా ఉంటుందో చూడండి. సో ఫ్రమ్ నౌ సపోజ్ ఫర్ ఎగ్జాంపుల్ maybe she may be drawing about 30,000 and definitely she will be awarded or elevated to the position of professor. Now already she is the vice principal and not less than 50,000 rupees she will be offered from this month itself. Okay, so I wish her all the best. She is a dedicated worker, she is a dedicated faculty. Okay, so I wish her all the best. బాబీ గారు నిన్న అమ్మాయికి PhD అవార్డు ఇచ్చారు ఆంధ్ర యూనివర్సిటీ.
Good evening to all. థ్యాంక్ యూ సార్. నేను 2009 అక్టోబర్ 22న ఈ కాలేజ్లో M.Pharm కంప్లీట్ చేసుకుని జాయిన్ అయ్యాను. సార్ 2010 ఆగస్ట్లో వచ్చారు. అప్పటి వరకు మాకు ప్రిన్సిపల్ ఎవరు లేరనమాట. సో సార్ వచ్చిన తర్వాత మమ్మల్ని బాగా గైడ్ చేశారు. మాకు అసలు ఏం తెలియదు చెప్పాలంటే, జస్ట్ ఏదో ఫ్యాకల్టీ అన్న జాయిన్ అయ్యామో ఏదో చెప్తున్నాం కానీ తెలియదు. సార్ జాయిన్ అయిన తర్వాత మమ్మల్ని బాగా గైడ్ చేశారు. ఆ తర్వాత రాఘవ సార్ జాయిన్ అయ్యారు. అయితే ఏంటంటే డైరెక్టర్ సారు ఒక ఫాదరు. ఫాదర్ ఎలా అయితే పిల్లల్ని మంచిగా ఉండాలని కోరుకుంటారు, అలానే డైరెక్టర్ సార్ కూడా, అలా మేడం కూడా, ఇద్దరు ఒక మంచి తల్లిదండ్రులాగా మమ్మల్ని చూస్తారు ఇప్పటికి కూడా. అలానే బాగా ఎంకరేజ్ చేస్తారు ఇద్దరు కూడా. తర్వాత ఇంతవరకు ఈ కాలేజ్లో ఇలా ఉన్నాము అంటే అది సార్ వాళ్ళే ఫస్ట్. తర్వాత రాఘవ సార్ కూడా ఒక బ్రదర్. మేము ఫస్ట్ 10 కదా సార్, మీరు 10 నుండి సార్ కూడా మాతో పాటు ట్రావెల్ అయ్యారు. మేము ఫ్యాకల్టీగా జాయిన్ అయ్యాం. ఆ తర్వాత మా బ్రదర్ అయ్యారు, ఇప్పుడు ప్రిన్సిపల్ అయ్యారు. అలా మీ అందరూ కూడా, స్టూడెంట్స్ అందరూ కూడా అంటే అలా చేసినప్పుడు తిడతాం, కానీ మీరందరూ మంచిగా ఉండాలని. సో ఇలా ఈ రోజు నేను ఎలా ఉన్నానంటే ఇది కాలేజ్, అలాగే మేనేజ్మెంట్, అలాగే మెయిన్గా డైరెక్టర్ సార్ వల్ల. అందుకే నేను చాలా సంతోషిస్తున్నాను. థ్యాంక్ యూ వెరీ మచ్.
ఓకే, ఆల్ ది బెస్ట్. థ్యాంక్ యూ, విష్ యూ ఆల్ ది బెస్ట్. అట్లాగనే మిగతా ఇంకా ఫ్యాకల్టీ ఇప్పుడు నెక్స్ట్ అప్కమింగ్ వుడ్ బి పిహెచ్డీస్ డాక్టర్ వుడ్ బి ఎడ్వర్డ్ రాజ్, వుడ్ బి డాక్టరేట్ ఎడ్వర్డ్ రాజ్. సో ఈరోజు ఈ ఫంక్షన్ అంతా సక్సెస్ఫుల్గా జరగడానికి కూడా కారణం ఎడ్వర్డే. సాయి పనీంద్ర ఈజ్ ఆల్సో దేర్. పనీంద్ర, నువ్వు రిజిస్టర్ పిహెచ్డీ. సో ఐ ఎంకరేజ్ మేడం, ఐ ఎంకరేజ్ ఈచ్ అండ్ ఎవరి ఫ్యాకల్టీ టు బి రిజిస్టర్డ్ ఫర్ ద పిహెచ్డీస్. ఎక్కడ ఏ యూనివర్సిటీ అయినా పర్వాలేదు. సో గెట్ రిజిస్టర్డ్ ఫర్ పిహెచ్డీ, డూ ఇట్, విల్ ప్రపోజ్. అతనికి ఆంధ్ర యూనివర్సిటీ రాలేదు, లక్కీ. ఆదికం నన్ను యూనివర్సిటీకి వచ్చింది. శైలి కాదు అమ్మాయి, ఇంకొక అమ్మాయికి అక్కడ వచ్చింది ఆంధ్ర యూనివర్సిటీ. ఎవరో షైనీకి, షైనీ షీ గాట్ సెలెక్టెడ్ ఫర్ ఆంధ్ర యూనివర్సిటీ అండ్ ఈ గాట్ సెలెక్టెడ్ ఫర్ ఏకేఎన్యూ. ఇట్ డజంట్ మ్యాటర్. సో అక్కడ అంతే, మాకు ఇక్కడ డ్యూయల్ రోల్. ఓకే, సో మనకున్న ఎక్స్పీరియన్స్, నాకున్న ఇన్ఫ్లుయెన్స్ తోటి, సో వీ కెన్ మేనేజ్ ద థింగ్స్. ఓకే, థ్యాంక్ యూ బాబీ గారు ఫర్ కమింగ్ ఓవర్ హియర్. ఓకే,
So now it's the time to felicitate Department of Pharmaceutics coordinators and the staff members. I request Bogireddy Sri Sweta to come on stage. The co-convener of the today's event. I request Director Garu to felicitate with the shield. షాల్ ఇవ్వండి, షాల్ ఇవ్వండి, ఉన్నాయి. ఒకసారి పంపండి.
ప్రతి ఈవెంట్ యంగస్టర్స్, faculty members, they come with a proposal. We wanted to conduct a seminar, national seminar. So unexpectedly international అయింది. సో యంగ్ ఫ్యాకల్టీ మెంబర్స్ ఓకే అడిగిన వెంటనే వీ హ్యావ్ టు ఎంకరేజ్, వీ హ్యావ్ గివెన్ ద ఆపర్చునిటీ టు కండక్ట్. సో విత్ ఇన్ స్పాన్ ఆఫ్ 1 month దే కుడ్ ఆర్గనైజ్ ది ఈవెంట్. సో నేను వాళ్ళ ఫాదర్ కనిపించి, మీలాంటి వాళ్ళ దగ్గర మా అమ్మాయి ఇంకా బాగా డెవలప్ అవ్వాలని చెప్పేసి ఈ వాజ్ ఆస్కింగ్. ఈ ఈజ్ ఏ లాయర్, అడ్వకేట్, కమ్యూనికేట్. ఎందుకంటే నేను వేర్ ఎవర్ ఇట్ ఈస్ దేర్, ప్రతిదీ కూడా మనం వాళ్ళకి చెప్పాలి, నేర్పించాలి. వాళ్ళు డెవలప్ అవ్వాలి స్కిల్స్. సో షీ కుడ్ ప్రూవ్ ఇట్. వాళ్ళ టీం ముగ్గురు ఉన్నారు, ఖతీజా, పూజిత, శ్వేత. వీళ్ళ ముగ్గురు కూర్చుని కలిసి పనిచేసి సో దే కుడ్ మేక్ ఇట్ ఎ గ్రాండ్ సక్సెస్. థ్యాంక్ యూ అండ్ విష్ యూ ఆల్ ది బెస్ట్. డూ మోర్. నువ్వు కూడా పిహెచ్డీకి తొందరగా రిజిస్టర్ చేసుకోండి. యూ హావ్ టు obtain the PhD.
One more news, sir. Today, just now, I speak with Sridhar Babu Garu. So we are planning to register Sri Shweta in PhD in a couple of months. Next, I request Kandavelli Jacob Isaac to come on to stage. Mr. Kandavelli Jacob Isaac, come on to the stage. ఉంది మీరు పంపించండి. Next, P. Kranti, possibility Kranti. Get ready. Next, P. Kranti. I request Muhammad Khadija Begum. First, Tota Jahnavi to come onto the stage. So I request Motukuri Tulasi to come onto the stage. Motukuri Tulasi. Thank you, sir, for felicitating Department of Pharmaceutics. So now it's time for declaration of the award winners for different posters and e-poster events. I request K. Devi to announce the prizes. I request Dr. Sridhar Babu Garu and P. Shailaja Madam, Associate Professor, to come on to the stage for delivering the prizes to the winners. So organizers. So third prize in e-posters goes to D. Vijay Kumar, AKRG College of Pharmacy. So the second prize for e-poster goes to M. Joshita, VV Institute of Pharmaceutical Sciences. M. Jyotika from VV Institute of Pharmaceutical Sciences. And first prize of e-poster goes to K. Supriya of Andhra University College of Pharmaceutical Sciences, Visakhapatnam.
For poster presentation, P. Sharon Lilly from Lydia College of Pharmacy, first prize. Second prize. For poster presentation, first prize P. Sharon Lilly, C. H. Chandini, D. Lakshmi. Team set, team members. Second prize goes to R. Haindavi from AKNU Tadepalligudem. G. Sirisha. Third prize goes to M. Kusuma from JITS College of Pharmacy. D. Jasmine. For oral presentation, first prize goes to D. Gayatri, VV Institute of Pharmaceutical Sciences. D. Gayatri. Second prize goes to E. Geeta Rani from KGRL College of Pharmacy. And third prize goes to K. Rajesh, Dr. CSN College of Pharmacy. felicitation for the evaluators. There are some consolidate prizes for the students. Consolidation prizes. For e-poster, consolation prizes. For e-poster presentation, R. Hema Sri from AKRG College of Pharmacy. Next one, STP Pranusha, Lydia College of Pharmacy. Next, Bhagya from JTIS College. JITS College. Next, Asha Jyoti from Andhra University, Visakhapatnam. A. Durga from JITS College. M. Suchaitra from KGRL College of Pharmacy. Nathaniel for Lydia College of Pharmacy. Madhuri from Andhra University. P. Madhuri. Now felicitation for evaluator Sheikh Rashid, M.Pharm, VV Institute of Pharmaceutical Sciences. Welcome, sir. Next one, N. Soni Priyanka ma'am from Department of Sutex, Jogaya College of Pharmacy. Please come on to the dais, ma'am. K. Venkateshwar Raju sir from Sri Vishnu College of Pharmacy. Welcome, sir.
Thank you, sir. Thank you, madam. So by this we completed our felicitation and prize distribution session. Now it's time for vote of thanks. Indeed, it a great honor and privilege to propose the vote of thanks on this memorable occasion. Let me first of all start by giving glory to the Almighty God for making today's occasion a resounding success. My heartfelt thanks to all guest speakers, delegates, director, principal, vice principal, head of the departments, teaching staff and non-teaching staff and students for successfully completing this first international seminar or conference at KGRL College of Pharmacy. Thank you one and all. So I request all the teaching staff members of KGRL College of Pharmacy to report to the stage for a group photo. So I request all the teaching staff. So make a call to all the teaching staff to assemble near the dais in seminar hall.